Kita kongsi dengan second screen. Okay. Itulah. Okay. Mungkin you nampak dekat screen? Nampak slide ke? Oh, you nampak? Sebab saya sini confuse dengan saya punya screen. Sebab saya banyak, banyak screen at the moment. Okay, so let's recap on what we have discussed last week. Okay, last Wednesday. All right, so can somebody recap? Can somebody share what you got from last lecture? Instead of about transcription, about the component, about the process, about the enzyme. Anyone would like to share? Let's recap before we continue with the mechanism, with the process. Anyone? Anyone would like to recap, to share about RNA polymerase? About transcription in prokaryote system? So what is promoter? What is coding, non-coding strand? What is operon? What is Tata box? What is -35 region? What is -10 region? Can somebody describe the structure of RNA polymerase? What does it mean by coenzyme? Haziq, mana Haziq? Haziq ada? Hai Haziq, mana Haziq? Haziq ada tak?
Di tandas doktor.
Okay. Alright. Okay, Eliana. Nurul Eliana kan? Do I pronounce your name correctly? Nurul Eliana. Anything that you want to share, anything that you want to recap, any important points that you got from last lecture. Okay, what is transcription? Okay. Nurul Eliana, pergi toilet juga ke? Afikah, Nurul Afikah Syahira. Saya
Yes,
tak ingat tau. Saya panggil you all tak respons ni.
Yes, Doctor.
Okay,
Can you hear
Afikah,
me?
anything? Yep.
I just can just describe about RNA polymerase, which is the structure. The RNA polymerase is consists of five subunits, which is contains alpha, beta, like beta prime, and beta, and another one is beta.
Okay.
So,
Alright, okay, thank you.
Okay. That's it. Thank you.
Alright, thank you, Afikah. Can I have Flora?
Yes, doctor.
Alright, anything you would like to share? You can describe what is sigma factor.
Sigma factor is to recognize the promoter for synthesis of initiation. Correct or not?
Alright.
I'm not sure.
Okay, that's correct. Alright, thank you, Flora.
Thank you, doctor.
Okay, Fariha. Fariha ada tak? Dia sebenarnya macam spot check lah, nak tengok whether you all ada ke tidak sebenarnya behind the screen. Kan? Kadang nama ada but you sendiri tak ada dekat situ. Fariha. Yeah.
I want to share about the transcription process. Is actually the synthesis, of RNA molecule using DNA strand as the template. So there will be a transformation of genetic information from DNA to RNA and then make functional protein.
All right, thank you, Fariha.
Okay.
At least you have something to share. Okay, how about Razik? Razik Irfan. Razik. Razik tak ada ke? Razik? Nope. Saya consider tak ada lah ni ya. Alright. Yang tidak respond, even though nama ada, but if you don't respond, if you are not response to the activity, so I assume you are not you don't join the lecture. Okay. Okay, Adriana Shakira. Okay, anything, Adriana?
Can you hear my voice?
Yep.
So I would like to share about the sense and anti-sense strand.
Okay. What is antisense strand?
This is from my understanding. So sense strand is then it has two strands, but only one strand that we use as template in the transcription. And the one used as template in transcription is sense strand. So another one, this sense strand is the complement DNA strand that is not used. So the one strand that not used for template called as anti-sense strand.
All right. Thank you, Adriana. So Adriana has explained about what the difference between antisense and also sense strand. Okay. Divyasri?
Yes, Doctor.
Hello. Okay. So anything you would like to share?
For the polymerase, I will talk about the polymerase. There are three types of RNA polymerase, which is RNA polymerase 1, 2, and 3. So the first one.
Okay.
So the first one is it synthesizes the precursors of most RNAs, whereas the RNA polymerase 2 it synthesizes the mRNA precursors, and the third one it synthesizes the precursors of 5 S of rRNA and the tRNAs and the variety of snRNA.
All right. Okay. Thank you, Divyasri. So basically, this multiple types of RNA polymerase only present in eukaryotic system. Okay, but for prokaryotic system, you only have one RNA polymerase. Okay. All right. Thank you very much. Mariam Sakina.
Yes, Doctor.
Okay. Anything you'd like to share?
I would like to share about catalogs and promoter.
Can you speak louder? Okay. Okay. You would like to share about data box?
Data box and promoter.
Okay.
But based on
Data box
my
and promoter.
understanding, data box will carry in a sequence that indicates where a genetic sequence that can be read and decoded.
Okay.
Okay, well, for the promoter, it's actually a region where DNA sequence that RNA polymerase can bind to initiate transcription.
Okay. Okay. So data box. So is there any specific region that we can identify data box in the promoter?
Yeah, like 35 region, 10 region.
Data box is -10. -10 region. Okay. -10 region is specific. Is conserved sequence for data box. Okay. -10 region. Okay. Thank you, Mariam. Okay. Tan Shin Er.
Yes, Doctor.
All right. Anything you'd like to share?
I'd like to share about recognition of origin.
Okay.
So about like there is operon, and then every transcription region, and then the operon includes some structural genes and
upstream regulatory sequence, and then we have a promoter, and then it's the it's the DNA sequence that RNA can bind, and then it is used to control the transcription.
Okay. All right. Okay. You have that. You have the point. Okay, but I think you have the point, but I think maybe you have to simplify it more. All right, about the you're talking about origin, correct? Betul tak? Origin kan?
Yes.
All right, okay. So thank you, Shin-er. Okay. Kenapa hanya ada 38, 39? You all semua berapa? 49 kan? Class rep. Berapa total class?
Dalam 49 macam tu.
49. Okay. All right, we'll take note. All right, so okay, thank you very much, everyone. So basically, it's an activity to recap. So at least I hope you still have something that you got from last lecture before we move to the next subtopic on the mechanism of the transcription. All right, so at least you able to describe what is RNA polymerase, what is the structure, what is the component. So what are the 5 components? So what are what is the role of each component involved in the RNA polymerase? So what is transcription itself? So what is it mean by sense, antisense? Okay, at least you have some idea, even though you are not understand yet. But I hope you manage, okay, to understand, to get some important points, okay, from last lecture to be used, okay, in this lecture, okay, in this subtopic. Okay, that's another activity to macam spot check jugalah, tengok you all ada ke tidak sebenarnya behind the screen. Okay, kadang-kadang ada 49, okay, but bila we conduct activity like this, okay, you are not around, okay, sebenarnya tak ada pun, okay, dekat belakang tu. Alright, so 39. Saya boleh ambil attending sekarang ke? Kejap ya. Okay. Right. So let's continue. Okay. We're going to discuss on the mechanism involved in the transcription process. Okay. 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 So as you know, okay, there are 3 general phases. Okay. That basically involved in either in the transcription process. Okay, we have initiation, elongation, and termination. Okay, and I already mentioned many times in application, you can see, you will understand about these three phases through activity of polymerase chain reaction. But this one we are going to look what does it means by these three phases: initiation, elongation, and also termination in the transcription process. Okay, so we have two different systems here. We have prokaryotic and eukaryotic system. Okay, and both obviously prokaryotic system, prokaryotic transcription process much simpler compared to eukaryotic system. Okay, for example, prokaryotic RNA polymerase can bind to the DNA template directly in the transcription process, but for transcription process in the eukaryotic system, it requires cofactors, it requires many other factors to bind to the DNA template, okay, for the transcription process. Okay, all right. Okay, so now let's look, okay, what happened in the three phases. Okay, in the initiation phases, in the initiation phase, it tells us about the recognition of the promoter by RNA polymerase and the binding of the promoter of the enzyme to the promoter, and this is where the transcription process initiated. Okay, and what it means by elongation phase. So this is the phase that the RNA strand is synthesized and continuously growing. Okay, for the termination phase, so it stops the RNA polymerase stops the synthesis. Okay, and the nascent RNA is separated from the DNA template. Okay. Right. Okay, so initiation. Okay, so it involves the binding of RNA polymerase holoenzyme, okay, to the promoter regions on the DNA. Okay, on the DNA. Okay, so if you still remember, okay, so what is promoter? So promoter, it has a sequence, okay, in which the transcription machinery binds and initiates the transcription. So basically, promoter, it exists upstream, okay, exists upstream of the genes that they regulate. Okay, although promoter vary among prokaryote genomes, but a few elements are conserved. Okay, there are few elements that are conserved, for example, at -35 and also -10 regions. Okay, so that are the regions where the sequence are highly conserved, meaning that it's similar in all bacterial species. Okay, what does it mean by conserved? The sequence is similar in all bacterial species. Okay. Okay, so -10 and -65 regions are the regions where the sequence are highly conserved. Okay. So the to recap about last lecture. Okay, -10 consists a sequence, also known as a -10 region. Okay, it has TA, AT. Okay, or we know as a Tata box. Okay, and okay, you need to know the sequence that the conserved sequence that code for -10 regions, and also you need to know what is the conserved sequence for the -35 region. Okay, so this -35 region is TTGACA. This is the sequence that recognized and bound by the sigma factor. Okay, okay. So once this interaction is made, okay, the subunits of the coenzymes, okay, will bind to the site. Okay. So this, okay, so you need to know, okay, what does it mean by consensus sequence, what is the sequence for -10 and also -35 regions. Okay, so in both prokaryotes and eukaryotes, purine ribonucleotide is usually the first to be polymerized into the RNA molecule. Okay. Okay, once the interaction is made between enzyme RNA polymerase and also to the promoter. Okay, the subunits, the sigma factor, okay, the sigma subunits of the enzyme will bind to the site and it will force off from the RNA polymerase. Okay, and this is where the first phosphodiester bond is formed. Okay, and then the coenzyme of the coenzyme will move along the DNA template to enter the elimination phase. Okay, let's look what happened to this. Okay, if you look at this diagram, if you look at this figure, okay, you know that RNA polymerase is a holoenzyme. Okay, holoenzyme. So it will sliding, okay, along the DNA template. Okay, it's sliding, it scan, it try to look for the specific sequence that they recognize to bind for the transcription process. Okay, which is at -35 region. Okay, once it's bind to this -35 region of the promoter, so it will form a close complex. Okay, form a close complex, and what happened here is, okay, there is an AT reach. Okay, dekat sini sebenarnya ada AT reach. Okay, if you, okay, -10 region, okay, TA, okay, TA, okay, or tatabol. So basically it's a AT reach region. Okay, AT reach sequence. Okay, sekejap ni. It's battery. Okay. Okay, so -10 region is a AT rich sequence, TA, AT. Okay, so this region basically facilitates the unwinding of the DNA template. We know that AT sequence, AT rich sequence, it doesn't have a very strong bonding. Okay, so that this AT rich sequence that facilitates the unwinding of the DNA template. Okay, unwinding of the DNA template, and so this is when the phosphodiester bond is formed. Okay, so the transcriptionation phase ends with the production of abortive transcript, which the polymers are approximately 10 nucleotides that are made and released. Okay. So once it forms a close complex, okay, AT rich facilitates the unwinding. Okay, at the same time, sigma subunit separates from the core. Okay, AT rich facilitates the unwinding. At the same time, sigma subunits, okay, falls off the RNA polymerase, so it's dissociated from the RNA polymerase. So this is when the phosphodiester bond is formed. Okay, so this is a situation that we can call it initiate the transcription process. Okay. Okay. All right. Okay. So basically, this sigma factor. Okay, the sigma factor of prokaryotic RNA polymerase recognize the consensus sequence in the promoter region. Okay, sigma factor is the one that recognizes the consensus sequence in the promoter region upstream of the transcription start site. Okay, so this is what happened here is the sigma subunit, okay, dissociate, okay, from the RNA polymerase after the transcription has been initiated. Okay. All right. Ada soalan on the initiation? Okay. Take note. Okay, we have at the moment discussing on the transcription process of the prokaryote system. Okay, so what happens during the elimination? Okay, what happens during the elimination? Okay, the release of the sigma subunit causes the conformational change of the coenzyme. What it means by that? Okay, so basically the elimination process begins with the release of sigma subunit from the polymerase enzyme. Okay, so the dissociation of sigma factor allows the coenzyme to proceed along the DNA template. Okay, so it helps, it facilitates, it proceeds with the synthesizing of mRNA in the 5' to the 3' direction at the rate of approximately 40 nucleotide per second. Okay, so now coenzyme can proceed along the DNA template. So it synthesizes the synthesized mRNA in the 5' to 3' direction. Okay, so as elongation proceeds, the DNA is continuously unwound. Okay, all right. So what is transcription bubble? Okay. Okay, so the transcription in either prokaryotes or in eukaryote system requires the DNA double helix to partially unwind, okay, in the region of mRNA synthesis. Okay, either in prokaryote or in eukaryote, so the DNA double helix need to be partially unwind. Okay, so the region of unwind the region of unwinding, DNA and unwinding is called as a transcription bubble. Okay, so there is a region of unpacked DNA. Okay, so the transcription bubble moves down the DNA, leaving the growing strand protruding synthesis from the bubble. Okay, all right, so there are 3' segments. So you can see the difference between the 3' and also 3' segment, also 5'. And okay, the 3' segment, okay, is it hybridized with the DNA template. So this one is the one that hybridized with the DNA template. For the 5' end, it sends out the transcription bubbles as the synthesis is processing. Okay, remember that the mRNA synthesis occurs from 5' to 3', okay, from 5' to 3'. So this is where the mRNA is synthesized, okay, from 5' to 3'. Okay. Okay, there are 2 types. Okay, so that's explain about elimination. Okay. So we have initiation, we have elimination. So it's a straightforward process for prokaryotic system. Okay, for prokaryotic system. Okay, now once a gene is transcribed, okay, the RNA polymerase needs to be instructed, okay, to dissociate, okay, from the DNA template and liberate to the newly made mRNA. Okay. Okay, so okay, now we have synthesized mRNA, so we need to tell the enzyme to stop. Okay, they need to dissociate. Okay, and we want to liberate the newly made mRNA. So there are 2 types. Okay, there are 2 types of termination signals. One is row-dependent termination, and also another one is row-independent. One is dependent, another one is row-independent termination. So what is row? Row is a protein. Okay, protein. Okay, it's you can say it's a protein base. Row-dependent termination is a protein base because row basically is a protein. So it's an ATP-dependent RNA stimulated helicase that disrupts the nascent DNA RNA complex. So this is this is a description for the role of a row. Okay. So basically for row-dependent termination is a termination process that controlled by rho protein. Okay, and this rho, this protein is tracks along behind. It tracks along behind the growing mRNA chain. Okay, dia ada je. It tracks along the mRNA chain. Okay, okay. So basically, so near to the end of the gene, okay, the polymerase encounters a run of G nucleotide on DNA template. Okay, it encounters a series of G nucleotide, okay, on DNA template. So as a result, rho protein collides, okay, with the polymerase, okay, and this interaction with rho protein, okay, releases mRNA from transcription bubble. Okay. All right. So interaction between rho protein and RNA polymerase, so it unwinds, so it release, okay, release the mRNA, release the RNA transcript. Okay. So now RNA polymerase and rho protein is dissociated. Okay. So now this is rho dependent termination. Okay, so it depends on the rho protein, and this termination is controlled by rho protein. Okay, how about rho independent termination? Okay, so this is RNA based. Okay, and this type of termination is controlled by a specific sequence in the DNA template itself. Okay. So it is depends on the sequence of the RNA transcript itself. Okay. So what is the termination sequence for the rho independent termination? So the termination sequence is a GC rich, okay, and also a series of uracil residues. Okay, so near the end of the gene, so basically it encountered a region rich in GC nucleotide. Okay, so once it found this GC rich, okay, you know that GC rich is the termination sequence. Okay, so once it's encountered a GC rich region, so mRNA falls back, okay, on itself. Okay, so the complementary CG nucleotide, it will bind together. So it will form a hairpin. Okay, hairpin. Okay, what does it mean by hairpin? Kita akan tengoklah. So what is hairpin? Followed by a stretch of U residues. Okay. Okay, so basically hairpin is a very stable formation. Okay, and the formation of hairpin, it will cause the polymerase, okay, to stall as soon as it begins to transcribe a region rich in 18 nucleotide. Okay, so the stem loop, okay, causes RNA polymerase to pause, and we know that the uracil residues is unstable. Okay, so it's very unstable, so it releases the RNA transcript. So this is how termination by rho independent termination. Okay. Okay, this is stem loop. Okay. We will see. So okay, what is stem loop disruption? Okay, the stem loop structure, basically it alters the conformation of RNA polymerase, so leading to the pause of the RNA polymerase moving. Okay, so it will cause it will reduce the stability of the DNA RNA, and it causes the transcription complex dissociated. Okay, so you can see there are two kinds of transcription termination in bacteria. One controlled by the rho protein, rho factor. Okay, and another one, okay, for rho independent termination, it will form a stem loop. Okay, so the formation of stem loop it gives a signal of the releasing of mRNA. So this is the storage of uracil residues, and this is a very unstable region. So it will release. It will permit the release of mRNA. So we have discussed on the initiation, elongation, and also termination in the transcription of prokaryotes. Okay, ada soalan? Tak? Any questions?
Doctor,
So, yep.
can you go back to the stem loop disruption?
Okay.
Can you explain the second sentence?
The competition of the RNA hybrid and the DNA RNA hybrid reduce the DNA RNA stability. Okay. Okay. I hope you can imagine this. Okay, this is the DNA. This is your DNA template. So you want, okay, in the transcription process, okay, we use DNA template as a template for the mRNA synthesis. Okay, so that's where DNA RNA. So what does it mean by DNA RNA? And also we have RNA hybrid, meaning that, okay, this is RNA hybrid. Okay, where this is the RNA. Once mRNA has been synthesized, and there is a region where the mRNA synthesis process is still occurs, okay, which they are using DNA template, DNA as a template. So it tells you the competition of the RNA hybrid between RNA meaning between mRNA itself, and the DNA RNA hybrid reduces the DNA RNA stability, because we at the end, okay, now is once it's formed at this stage, when is reached this stage, we know that mRNA has been we can assume RNA synthesis has been almost complete, but still we need this termination to give a signal to the RNA polymerase to stop the mRNA synthesis process. We want to stop the transcription process. We want to stop the conversion of DNA to RNA. Okay, DNA RNA, we know that once, okay, if there is no termination process occurs here, there will be a transcription of DNA to RNA. Is still ongoing process. So that's why there is a competition between RNA and also DNA RNA. Okay, so we know that once because stem loop, even though it alters the conformation, but it is a very stable conformation. So that's why RNA hybrid it will reduce the DNA RNA stability. Because once there is a formation of stem loop, it will alter the conformation, and it facilitates the dissociation of RNA polymerase from the transcription complex. Okay, Danu? Danu, you okay?
Okay, Doctor.
All right. Okay, any other questions?
Doktor, saya sebenarnya tak faham yang RNA hybrid tu, Doktor, still tak faham.
Okay, RNA hybrid, basically, okay, now at this stage, we have synthesized, there is a nearly complete synthesis of mRNA. So that's why RNA. So everything have been transcribed. Okay, DNA has been transcribed to mRNA. So it's just a term. Okay, RNA to show that there is a complete synthesized mRNA synthesis process. So because dekat sini kan kita elongate, we want to transcribe mRNA from DNA. So it's just a term to show that we are transcribing RNA from DNA. But at certain stage, we need to tell the RNA polymerase to stop synthesize mRNAs. So that's why we have termination process. Okay, we need to tell RNA polymerase to stop transcribe the DNA to RNA. Okay, that's why we have termination process. So RNA hybrid is a term to tell us that we have enough mRNA transcribed from DNA. But still, kalau kita belum ada, if we don't signal the RNA polymerase to stop the transcription, there is still ongoing transcription process. That's why ada DNA RNA, because we know the transcription process is we require DNA as a template. Dia hanya term, okay? You don't think because it's a simple process to transcribe DNA RNA. Kita hanya nak dapatkan mRNA in this reaction. Okay, dekat sini. Okay. Do not make yourself confused with the term hybrid. What is that mean by RNA and also DNA RNA. Okay. Kena just
maksudnya dia tak stop sendirilah doktor at the codon. The codon doesn't stop
that's why
sendiri.
That's why that's why the determination process. There are two kinds of process. Either it depends on the raw protein or independent termination. Okay, kalau independent termination, it has their own sequence. Dia ada dia punya termination sequence. It depends on which termination process that they are taking. Okay, if they are using row dependent, so they are going to have this. Okay, because if they have that raw protein, raw factor that tracks along the mRNA, so they are going to have termination based on raw protein. But if they don't have raw protein along the way, the mRNA synthesis process, so they are going to use this RNA independent termination. So the termination process will occur once they encounter this GC-rich sequence. Okay, GC-rich sequence. So they will form a hairpins. Okay, stem loop. Okay, hairpins or stem loop. Okay, and the series of uracil will help to dissociate, okay, the RNA polymerase from the complex. Okay, this is how the transcription process is terminate. Okay, still kena ada termination process. Kalau tak, the transcription process is ongoing process. So that's why kita punya system is very smart. They can signal. Okay, that's enough. Okay, we don't have that much. Okay, we need to stop the process so that this mRNA can undergo the next step, which is translation. Okay, now kita perlu ada mRNA. Okay, mRNA is not function without being translated to the protein. So this mRNA, once it has a very complete sequence of mRNA, so it needs to be so the translation process needs to be stopped, so it can undergo translation process to form a protein. So that protein will be expressed. Okay, so that all the role of the protein can be expressed as it is. Okay, okay, Nadira.
So apa kaitannya dengan stability DNA RNA?
Okay. Alright, stability. Okay. Sekarang ni dekat what does it mean by DNA RNA stability? Okay. Sekarang you imagine we have this stem loop. Okay. Saya tunjuk ni. Once it's formed this stem loop, okay, selalunya DNA dia punya mRNA punya structure dia akan form linear. Okay, linear sequence, right? Correct. Okay. So once it form this hair loop, once it encounter this series, dia akan form hair loop. This hairpin loop ataupun stem loop. Obviously, it will alter the conformation. Correct? It will alter the conformation of the sequence. Okay, it will alter the conformation of the RNA polymerase. Okay. Okay. At the same time, it has this unpaired sequence of uracil residues. There are the series of uracil residues. Okay, at the end of the stem loop. Okay. So once kalau dia alter the conformation, it will reduce the stability. Okay, it will reduce the stability of the complex. Kan, apa sajalah contoh kalau kita punya DNA, kalau berlaku mutation, dia akan perubahan satu nucleotide pun, it will affect the stability. It will change the conformation of the structure. Betul tak? Okay, contoh dalam structure DNA RNA dekat carbon number 2. Okay, replacement between hydrogen dengan hydroxyl group, it will give you totally new molecule. Okay, same goes to this process. Okay, daripada linear sequence, once it form a stem loop, it will alter the conformation and it will affect the stability. Okay, lagi-lagi ada this series of uracil residues. This series of uracil residues, it will cause, we know that uracil ke, AT-rich ke is very unstable. So it will lead to the dissociation of the RNA polymerase from the complex. Because they alter conformation. Biasa sequence, linear sequence of mRNA ke, DNA ke kan, kita ada sequence about hidup and everything. Once it forms stem loop, okay, dia bentuk macam tu, so it alters the conformation. So alters the conformation, dia akan reduce the stability of the DNA RNA ke RNA ke. Okay. Okay. So hairpin loop, dia akan determines the independent termination. Alright. Okay. I hope it helps. Ada soalan lain? Before we move to eukaryotic transcription. Okay, so transcription of eukaryotic gene is far more a complicated process than prokaryotes. Okay, now I hope you able to understand, you're able to at least outline the transcription process for prokaryotes, so that you can see the difference between prokaryotes and eukaryotes. Okay, later saya punya tips kalau if you want to do your own revision, okay, in one paper, differentiate. Okay, you buat comparison between transcription prokaryotes and also transcription of eukaryotes. So you can see the difference. You can see what are the additional enzymes, additional steps, okay, involved dalam eukaryote transcription of eukaryote. Okay. So this is what happened in the eukaryotic transcription. Okay. Okay. Jadi kita tengok participant dah jadi 47, 46 lah. Alright. 10 o'clock baru 47. Alright. Okay. So in general, so this is what happened in the eukaryotic transcription. So still it can be divided into 3 phases: initiation, elongation, and termination. So basically, in general, process is still sama. Okay, initiation, we know that it involves the recognizing of initiation binding site by sigma factor at the promoter region. Okay, so that's how the initiation of transcription occurs. Okay, so RNA polymerase recognizes the recognition sites, causing it to bind to the promoter. Okay, so RNA polymerase then separates the DNA into single strands so that the template strand can be read in the 3K. So later we will look on the additional steps involved in the initiation or elongation or termination in eukaryotic system. Okay. All right. So for elongation, basically sama. Okay. Pre-mRNA nucleotides are quickly packed with their complementary bases, which correspond with the template strand of DNA. Okay. Basically sama lah. It's about growing the mRNA strand. Okay. Is an elongation of mRNA strand. All right. Move from the 5' to 3' direction. Okay. For the termination, still in the eukaryotic transcription process, we still need termination step. Okay, to tell the RNA polymerase to stop, and we need to release the RNA. Okay. Okay, let's look in detail what does it means by initiation. Okay. Okay, in general, okay, transcription initiation needs promoter and upstream regulatory regions. Okay. Okay, we need promoter. You already know what is promoter, and also upstream regulatory regions. We need this -35 region. We need -10 region. So that's what it means by upstream regulatory region. Okay, -10, -35 region. Okay, and other than that, you see there are cis-acting elements that are the specific sequences on the DNA template that regulate the transcription of one or more genes. Okay, we are going to look at what is meant by cis-acting element. Okay. Okay, you can see on look at this diagram. So basically, this what does it looks like on the eukaryotic system for the promoter region too. Okay. Jadi DC box. Okay. Okay, it's much more complex compared to minus complex eukaryotic system. Okay, later we will discuss what is exon, intron. Okay. 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 Apart from promoter, apart from all the upstream regulatory region, we have in eukaryotic system we need these transcription factors. Okay, so in eukaryotic system, RNA polymerase does not bind to the promoter directly. Okay, because RNA polymerase 2, okay, kan dalam eukaryotic system we have 3 different RNA polymerase, and RNA polymerase 2, the one that involved in the initiation steps, okay, it needs to, it associated with 6 transcription factors, which is transcription factor 2A to H. All right. Ada 6 there are 6 transcription factors. Transcription factor 2 from A to H. A, B, C, D, A, B, C, D, E. Sekejap. A, B, kita ada C A, B, F, E, H. Okay, we have A, B, D, E, F, H. Okay. All right. So what is trans-acting factors? Tadi cis-acting elements kan. This is trans-acting factors. Are the proteins that recognize and bind directly or indirectly cis-acting elements and regulate its activity. Okay. Okay, do you know what does it mean by cis and trans? Ada yang tahu tak? Cis and trans in terms of the conformation. Tahu tak? Cis and trans. Okay. Okay, in terms of structure, conformation dia. Okay, anyone? Siapa yang boleh ingat? Okay, biasa, okay. Siapa yang biasa kalau nampak dekat any, especially the T lah kan, dia akan tulis 0% trans fat. Siapa yang notice? And other foods jugalah. Why they highlight that their products have 0% trans fat? Why don't cis fat? Yang tu nanti sebenarnya kita akan discuss dalam lipid sebenarnya, lipid metabolism. But at the moment now, I would like to know whether you can differentiate what does it means, okay, in terms of the conformation, in terms of the structure between cis and trans. Nanti cari ya, because later dalam next semester kita akan discuss, okay, dalam lipid metabolism. Okay, what is trans fat? Okay, why is it? Well, in the industry, they want to highlight that their product contains 0% trans fat. So what is the significance of that? Okay, what does it mean by 0% trans fat? Okay, how does it helps? Okay, in the food security and how it helps in contributing a good diet to the people. All right, so go back and check, okay, what is meant by cis and trans in terms of the configuration, in terms of the conformation of this structure. Okay, because you jumpa kat sini cis. Now, trans-acting factors are the proteins that recognize and bind directly or indirectly to the cis-acting elements. If you have trans, it helps, okay, cis-acting elements and regulate its activity. Jadi dah jumpa term-term ni, make sure you understand what that means by cis and trans. Okay, so this are the table that tells you about the function of the general transcription initiation factor. So all these 6 initiation factor, kita ada A, B, F, E, H, and also D. All right. Okay. TF2D and TDP is a tetrabox recognition. Okay, that positioning of tetrabox in A around the F2B and polymerase 2. All right. So TDP is a tetrabox binding protein. So it forms a complex with transition factor 2D. Okay. And also this one is a co-promoter recognition, okay, and it involves in the positive and negative regulation, okay. Transcription factor 2D and also transcription TAF, okay. Okay, transcription factor 2A, it helps to stabilize the TBP binding, okay. Transcription factor 2B, it involves in the recruitment of polymerase 2, and transcription factor 2F, okay, is also the start site recognition for polymerase 2. All right, okay, transcription factor 2F is a promoter targeting of from polymerase 2. For E, it helps to recruit transcription factor H, okay, and modulation of helicase, CPPase, and kinase activities. For transcription factor 2H is a promoter melting, promoter clearance via phosphorylation of CTD. Okay. Okay. Now let's look what happened in the pre-initiation complex. Okay, pre-initiation complex. So, okay, what does it mean by pre-initiation complex? So we have this promoter that contain a tata box. Okay, promoter dia ada tata box. Okay. So tata box binding protein, it recognize the tata box. Okay, okay. Kat sini. Okay, tata. Okay, transcription factor 2. Okay, this complex. Okay, this complex. Okay, it recognize the tata box of the promoter. So that tata box formation. So basically, first thing first, what happened is this molecule. Okay, the tata box binding protein. Okay, this TBP subunit of the F2D. It recognizes the tata box, so it helps to initiate the assembly of a transcription complex. Okay. All right. So now TBP of transcription factor 2D, it recognizes the tata box, so it binds to tata box. So what does it mean by binding to tata box? So it initiates the assembly of a transcription complex. Okay. First, okay, you dah tahu dah. Banyak-banyak dalam this 6 transcription factor, which one? Okay, starts the initiation, the pre-initiation complex. Okay. Next, okay, next proteins to enter are transcription factor 2A and transcription factor 2B. Okay, which they bind to transcription factor 2D, which they stabilize the complex. Okay, it stabilize the DNA transcription as a transcription factor 2D complex, and they recruit now polymerase 2 in association with transcription factor 2F. Okay. Okay, next protein to enter is A and B, which they bind to D. Okay, they bind to D and tadi tu. So now it's stabilized. So this binding stabilize the DNA, the F2D complex. Okay, and now they recruit RNA polymerase 2. Okay, to bind to transcription factor 2B. Okay, and next, okay, so transcription factor 2B serve as the bridge. Okay, they serve as the bridge between the tata bound TDP and also polymerase, and helps to place the active center of the polymerase in the correct position to initiate the transcription. Okay, now F and E. Okay, B, they serve as a bridge. Okay, serve as a bridge so that it helps to place the polymerase in the correct position to initiate the transcription. Okay, then F and E helps to unwind the DNA. Okay, okay, with the help of helicase and ATPase, so it requires ATP hydrolysis. Okay, and one of the last transcription factor to be recruited to the pre-initiation complex is transcription factor H, okay, which plays an important role in promoter melting, and it signals that it's a completion of pre-initiation complex. Okay, so for this slide, so basically you have to know the sequence. Okay, this is the sequence assembly for the pre-initiation complex. Which transcription factor comes first, and the role of each transcription factor. Okay, so which one comes first, which protein comes later, and which one is the last protein to enter for the completion of pre-initiation complex. So So you need to know the sequence and also the role of each factor. Okay. Okay, by looking at this diagram, you can understand the sequence of the formation of the initiation complex. Okay. All right. So this is another diagram to explain. Okay, on the formation of pre-initiation complex. Okay. Okay, you can just look. Okay, either by understanding the process by looking at this diagram or this diagram. Okay. All right. Okay. Okay, now we know that transcription factor 2H, okay, this FH helps in promoter melting, okay, and the completion of PIC. So basically, this step requires a hydrolysis of ATP, okay, and is mediated by the FH. And what is mean by hydrolysis, ATP is phosphorylate the CTD, okay, the C-terminal domain of RNA polymerase. Okay, so once it's phosphorylate the C-terminal domain of RNA polymerase, all right, so that so all the transcription factors falls off from the PIC during the elongation phase, and only RNA polymerase can move toward the downstream, starting the elongation phase. Okay, so this is how they enter from initiation step to elongation phase. Okay, now they want to give, so all the transcription factor need to be dissociate from the PIC, so that only RNA polymerase can enter, can move toward the downstream and starting the elongation phase. Okay. Alright, ada soalan? Nope. Ada soalan tak?
Doktor,
Yes.
kenapa kena promoter melting tu?
Kenapa kena promoter melting? So what does it mean by melting here? So we so we want to enter, okay, now once it binds, so this is pre-initiation complex, formation of pre-initiation complex, meaning it tells you how all the truncation, it explains how all the transcription factors helps in binding of the RNA polymerase to the promoter region, correct? But dalam eukaryotic system, it involves 6 different transcription factors. So this is pre-initiation complex, meaning that the binding of RNA polymerase to the promoter, but it involves 6 different transcription factors. So it tells you the transcription factor now, transcription process now can start and begins. Okay, but for the elongation phase, we need only RNA polymerase to enter the elongation phase, meaning only RNA polymerase can move down to the upstream, to the downstream. And ingat, you need to remember this promoter is located at the upstream region. Okay, you punya minus 35, you minus 10, all located at the upstream. So for the elongation phase, you punya only RNA polymerase can move down towards the downstream. Okay, can move down to the downstream of the DNA template. Okay, so that kat sini we need the process of promoter melting by the hydrolysis of ATP by the phosphorylate of the C-terminal domain. So we need all these transcription factor need to be fall off from the promoter. So we want to remove everything so that only RNA polymerase can move towards for the elongation phase. Can synthesize RNA, can synthesize mRNA. Okay, only RNA polymerase can synthesize RNA. Okay, Tanu? That's what it means by promoter melting. So promoter, we know that it's for binding, okay, to start to initiate the transcription process. Once it's bind, once it initiate the transcription process, so it will enter the elongation phase. So that's why only RNA polymerase can move towards to the downstream so that it can synthesize mRNA. So that's why the other phosphorylation of C-terminal domain, we want to all other factors, all the promoters need to be removed from the RNA polymerase. So how it being removed by promoter melting by RNA by phosphohydrolysis of ATP. And basically, this transcription factor H that helps in the promoter melting. Okay, this transcription factor H that play a role in promoter melting by catalyzing the phosphorylate of C-terminal domain of RNA polymerase. Okay, by the hydrolysis of ATP. Sebab kita now hanya nak RNA polymerase je yang move towards to the elongation phase for the synthesis of mRNA. We don't want any promoter transcription factors yang banyak tu kan. So once we form the pre-initiation complex, so you need to undergo this phosphorylation of RNAP to remove all factors lah. Okay, alright. Soalan lain? No? Okay, dah 10:30. Okay, saya kena laju kot. I want to finish everything by today. Elongation. Okay, elongation sama. Okay, sama seperti dalam prokaryote. Okay, is similar to that of prokaryote. Okay, is about growing of mRNA strand. Okay. Okay, dia sama je sebenarnya. What makes the process different from prokaryote is the termination process. Okay, eukaryotic genes contain a poly A signal. Okay, located downstream of the last exon. Tak ada capping kat sini. Okay, dekat capping at 5' end also at 3' end. Okay, dekat tail dia. Okay. So this signal is used to add a series of adenylate residues during RNA processing. Okay. Okay, RNA polymerase II reaches the polyadenylation sequence and releases the pre-mRNA. Okay, this is a poly A signal. Okay, so this is how termination occurs. Once RNA polymerase counter this polyadenylation. So it helps to release the pre-mRNA. Okay. All right. Okay. You can see this diagram. Okay, you can see this diagram. Okay. What does it mean by capping? And also tailing. Okay, tailing at 3' end. Okay, what does it mean by exon? What does it mean by intron? Okay, so this is just termination sites. Okay, all right. Nanti sekejap. Okay, so in eukaryotic system, one thing that make eukaryotic system is much complex because it has this post-translational modification. It's a process that gives an advantage to the eukaryotic system. Okay, is a process that involves in translating mRNA to DNA. It's a process, nama pun post-translational modification. So it's process to modify. It can modify. It can modify your DNA, meaning that it helps to encounter any problem during the translation process. Okay. So what does it mean by post-translational modification? Dalam prokaryotic system, there is no post-translational modification. Okay, it is a very unique system. Okay, it offers a lot of advantages, okay, to the eukaryotic system by having this process. Okay. Okay, the next one RNA. Okay, once we undergo all the elimination process, we now have this primary transcript of RNA. And this primary transcript of RNA needs to be modified to become functional tRNAs, rRNAs, and also mRNAs. Okay, and this modification is critical to eukaryotic systems. Okay, all right. Okay, so how does this modification being carried out? Okay, so modification of mRNA includes keeping at the 5' end, tailing at the 3' end, or also splicing of coding sequence. Okay, keeping at 5', tailing at 3', and also splicing. Okay, so these are the three modifications. What does it mean by modification? These are the three steps involved in the modification of mRNA. Okay. Okay, what it means by keeping at the 5' end. Okay, keeping at the 5' end. Okay, so this step occurs in nuclei. Okay, which 7-methylguanosine is added to the 5' end. Okay. Okay, the cap structure of mRNA will be recognized by the cap binding protein, which required for the translation, and the capping occurs prior to the splicing. And so what is the role of this capping? Okay, so it helps to protect the 5' end from degradation, okay, in the nucleus. Okay, so that's 5' capping. Okay, so 5' cap is added, okay, to the beginning of the RNA transcript. Okay. So basically it helps to protect from the breakdown, okay, from the degradation, okay, from the degradation. Okay, and it also helps the ribosome to attach to the mRNA and start reading to make a protein. Okay, tapi one of the important keyword, it helps, okay, it protects from the degradation. Okay, so this is what is meant by capping. Okay, so this is capping, okay, in mRNA. Okay, two capping processes occur. You can have a look at this diagram. Attachment of GMP residue at the 5' end and methylation at N7 of guanine and at the ribose of 200. So, okay, there are many. It occurs at the beginning of the RNA transcript. Okay, at the 5' end. Okay, capping by GMP residues. Okay. So what does it mean by tailing at the 3' end? So tailing by polyadenine. Okay, and it occurs in the nuclei. So what it means by poly-A tailing? Okay, more than 200. Okay, adenosine nucleotides are added to the 3' end by polymerase to create a polychain. Okay, okay, so it helps to stabilize the mRNA by increasing resistance to cellular nucleases. Okay, so basically it helps to protect, okay, the RNA, the mRNA. Okay, by having something at the end, at the beginning and also at the end of the mRNA transcript. Okay, kalau at the beginning we call as a capping. Okay, kalau at the end of the RNA transcript we call as a tailing by the adenine. Okay, all right. So what is splicing? Okay, what is splicing? Okay, so once we have this mRNA transcript, kita ada capping, we have this tailing. Okay, but the mature mRNAs that we are going to produce is much shorter than the DNA templates. Okay, so that's the structural genes are composed of coding and non-coding regions that are alternatively separated. Okay, okay. Normally, the non-structural genes we have all this our coding regions, something that we want to be translated, and also non-coding regions. Okay, and this is also known as an exon and also intron. Okay, so what is exon? So basically this is the structure of the pre-mRNA in what does this the sequence of pre-mRNA looks like. So for the pre-mRNA we have this exon, we have this intron, and so you can see that we have a sequence of combination between exon and intron in the pre-mRNA before it processed. Okay, so what is exon and intron? Exons is a coding sequences that appear on genes and primary transcript and will be expressed to mature mRNA. So basically exons is the coding regions. Okay, this is the coding sequence that you want that going to be expressed as to a matured mRNA. So for introns, okay, this is the non-coding sequences that are transcripted into primary mRNAs and will be cleaved out in the later splicing process. So introns is a non-coding sequence, is something that we need to remove. Okay, we don't want that. Okay, and will be cleaved out in the splicing process. Okay, so introns are cut from the gene and the exon fragments are joined, okay, spliced to produce mature mRNA. Okay, so let's say this is the, okay, this is your DNA, it undergo transcription and undergo all this keeping, all right, and all this, it undergoes splicing, okay, cleavage of the introns, okay, and now this example of the modification of a primary transcript begins by the capping at the 5' end. It depends, okay, there is a process of, we start with the capping, okay, followed by tailing, okay, end up by the it caps, okay, the 3' end with poly A tail, okay. So for the splicing, okay, introns are removed from the primary RNA transcript while it is still in the nucleus. Okay, introns are spliced out by RNA protein hybrids called spliceosomes. Okay, so basically, this steps we cleave out the intron. Okay, we cleave out the introns so that the remaining ends are attached to that. The final RNA consists of exons only. All right. Okay. Okay, so for summary, RNA polymerase produces a primary transcript and exact RNA copy of the gene. A cap is put on the 5' end, and the RNA is mutated and poly A is added to the 3' end. Okay, our introns are spliced out at this point. The RNA can be called messenger RNA. So, so this is how it looks like after the pre-mRNA processing, after it undergo the addition of introns and splicing of exons. Okay, splicing, it joins the exons together to form RNA that we want. So then it cuts, okay, the RNA by polyadenylation. Okay, then it can undergo a process to produce mature mRNA. All right, so that's it for transcription. Any questions? We have another 5 to 10 minutes. You all ada kelas ke lepas ni? Ada kelas ke lepas ni? Kelas, awak ada kelas ke lepas ni? Assalamualaikum.
Kelas bahasa doktor. Pukul 12.
Ya Allah, you all ni memang tak boleh respons ke? Okay, oh tak ada kelas, 11 tak ada kelas. Okay, alright. So, okay, saya baru baca you punya questions in the chat box. What is the difference between the Tata box and Putno box? Okay, try to find what is the difference between, okay, I'll give back that questions to you. Okay, the simple questions. Simple je questions tu. Okay, I'll give back that questions to you. Try to find out. Nanti saya tanya next class. Okay, thanks Danu for the questions. Okay, two simple, just straightforward questions. Okay, so that's why I pass it back to you and we discuss next for this day. Okay, ada soalan? Okay, if there is no questions, okay, nanti saya preparekan soalan. Mungkin hari Rabu, saya rasa saya completekan, saya settlekan dulu semua lecture. And hopefully by next week, if we still have time, kita boleh buat some tutorial. Okay, we can have some activity or discussion. Alright, for this topic and also transcript for the replication. Okay, boleh? Lah, boleh.
Doktor.
Ya Allah, kesian saya tahu, saya cakap seorang-seorang. Doctor, for presentation, can we use Malay as our catchy title? Depends lah tajuk apa. But it's advisable. Tengoklah. Can you direct DM me, message me? Oh, Dr. Dina. Saya kena tengok juga kan. Alright. So, thank you everyone. Conducted in online or face-to-face. Next week kan? Waktu lab. Alright, so we have 3 hours. Okay. If we stick on time,